Mendelian inheritance. Uh, Actually, what do you mean by inheritance? Inheritance is nothing but the transmitting characters from parents to children is called the inheritance. Those are the characters, either it may be anatomical or morphological, either it may be color or it may be anything. Those characters which are present in the parents have to be transmitted to the next generation. But here the question is, how do these characters are transmitted to the next generation? uh, John Gregor Mendel, scientist in genetics, father of genetics, he conducted his experiments on the Physum sativum pea plant, different characters in the pea plant he observed, and the mode of inheritance is also recessed by him, and he proposed some principles: the law of dominance, law of segregation, and law of independent assortment. These are the three principles proposed by John Gregor Mendel after conducting the experiments on the pea plant or Physum sativum in 1900s. So this is called those principles, and that inheritance is called the Mendelian inheritance. What do you mean by Mendelian inheritance? Means inheritance of traits or characters from the parents to children. Means inheritance of traits or characters from parents to offsprings or next generation. That is called the Mendelian inheritance, which are following the Mendelian principles. For genes and alleles uh, or factors follow fundamental rules while transmitting characters to the next generation. It means genes and alleles. Actually, John Gregor Mendel did not know that about the genes and alleles. Later, those were named as the genes and alleles, but the Mendel called them as factors. Those factors which are present in the parents can be transmitted to the next generation. Those were called as the factors, but now we are calling them as the genes and alleles. These genes and alleles or factors follow fundamental rules. What are the fundamental rules proposed by this John Gregor Mendel? Means those are the principles: law of dominance, law of segregation, and law of independent assortment. Those which we are going to be discussed in this video lesson. So follow fundamental rules while transmitting characters to the next generations. So this is the main topic in the. So what are the three principles? What are the main key points in the Mendelian inheritance? Those are the Mendelian principles. One is the law of dominance, law of segregation, and another one is the law of independent assortment. So Mendel uh, conducted one by taking one character in the pea plant, for example, tall or dwarf or white color flowers and uh, pink color flowers, like that. One character he was choose and conducted his experiment. That was called as the monohybrid hybridization. In this monohybrid hybridization, uh, F1 and F2 generations were observed. From these observations, he proposed this law of dominance. Law of dominance means in two alleles, one allele is expressed and another one allele is the masked. Means only one allele is dominant, allele is expressed. That is called the law of dominance. But the law of segregation means during the gametes formation, uh, in sexually reproducing organisms, during the gametes formation, the alleles present on the two chromosomes can be enter into the alleles. This are means the two alleles which are present on the two chromosomes at same locus can be enter into the gametes separately. That is the segregation. Segregation is nothing but dividing, separation. So allele separation is nothing but the law of segregation. And from the dihybrid hybridization, he proved this law of independent assortment. Those genes or alleles present on the same chromosomes are also enter into the gametes without disturbing. Those are the three principles: law of dominance, segregation, and independent assortment, which are the important principles proposed by Mendel in the Mendelian inheritance. So means in sexually reproducing organisms, the characters present in the parents can be transmitted to the next generation by following these three rules: law of dominance, segregation, and independent assortment. So John Gregor Mendel conducted his experiments on the pea plant. Uh, he chose actually 7 characters in the pea plant. Those 7 characters are uh, round seeds and wrinkled seeds. Round seeds means those are round in shape. Wrinkled means somewhat wrinkles present on the seed. So round seed and wrinkled seed. Then another character is the yellow color seed in some green color seed. Inflated pod color are constricted. So the pod the pod is smooth, but here the constricted pod is. These two are contrasting alleles. This row, the alleles present in this row are contrasting alleles to these alleles. So this is inflated. Next one is the pod color, green pod color and the yellow pod color. Next one is the flower, purple color flowers and white color flowers. And another character is the axial flowering, means the branch flowering on the side branches. So the contrasting character is terminal flowering. And another last one, seventh character or trait was tall plant and dwarf or short plant. These are the 7 characters he chose in the pea plant or Physum sativum. So these round, yellow, inflated, green, purple, axial, tall, these characters are contrasting to these characters. These alleles are contrasting to these alleles. So as well as genes. So um, the allele which can determine the round shape, and the allele which can determine the wrinkled seeds are contrasting and located on the 2 chromosomes at same locus. So those are determining one character. Either it may be wrinkled character or it may be round character. So these 2 alleles are participating in determining one character. These 2 alleles are present on the 2 chromosomes at same locus. Means we know that the alleles means 2 genes which are located on the same, on the 2 chromosomes at same locus and which can determine same character. Those are called the alleles. So these 2 are alleles which can determine one character, that is the shape of the seed. These 2 alleles can determine the color of that seed, either it may be green or it may be yellow. These 2 alleles can determine the nature of that pod, whether it is inflated, whether it is constricted. These 2 alleles can determine one character, that is that color is the pod color, either it may be green color or it may be yellow color. And these 2 alleles can determine the color of the flower, either it may be purple color or it may be white color. And these 2 alleles can determine where the flowers where the flowering take place in the plant, either it may be axial flowering or it may be terminal flowering. These 2 alleles can determine whether the plant is tall or it may be short or dwarf plant. These are the characters chosen by John Gregor Mendel in this Physum sativa. So another thing is these alleles which are located in these boxes, top boxes, these alleles, round, yellow, inflated, green, purple, axial characters, these are actually dominant characters observed by John Gregor Mendel. These are the wrinkled green, constricted yellow, white, terminal, short or dwarf plant are recessive characters, recessive genes or alleles. So these are the dominant alleles what he observed. These are the recessive alleles. So what is dominant alleles and what is recessive alleles means? Dominant alleles, what are they? Dominant alleles can determine the traits or factors. Those are round seeds, these round seeds, yellow seed, inflated pod, green pod color, purple flowers, axial flowering, and the tall stem. So these are the recessive characters. So dominant characters are expressed, recessive characters are hidden or masked by the dominant alleles. So we will discuss very clearly. So before going into the principles and Mendelian inheritance. Now I would like to discuss some of the terminology used in this topic. So one is the phenotypes. Phenotypes are nothing but the physically appearing characters. For example, external characters or morphological character. Means in the previous slide, what is the morphology? These are all morphological character, whether the seed is round in shape or in wrinkled. These all are the morphological characters or phenotypic characters. So these are called the phenotypic characters, physically appearing characters. It may be round, or it may be yellow in color, or it may be tall, or it may be dwarf. Those are the physically appearing characters are called the phenotypes. Those ratios which are formed in a particular generation are called the phenotypic ratios. Phenotypes, what do we, for example, tall, dark, white, yellow, external characters, green. These are the phenotypes. So another one is the genotype. So genotypes is nothing but genetic makeup of the genes or alleles. So for example, a tall plant is there. Tall plant means what is it? Its phenotypic, uh, its phenotype is tall. So that is physical appearance. But what is the genotypic ratio? A genotype of the tall plant means either it may be capital T, capital T. If it is the dwarf plant, it may be small t, or it may be in heterozygous condition, it may be tall. So what is the genetic makeup of the plant or organism is called the genotype. So phenotype is the external characters or physically appearing characters, or genotypes are the genetic makeup of the genes. And another one is the dominance. What do you mean by dominance? Dominance means the allele which can express and which can mask the other allele character are called the dominance. So expression of one allele by masking another means expression of one allele. For example, T allele is expressed by masking another allele. By masking the T allele means expression of one allele, capital T, by masking the another small t allele. Means here capital T allele is the dominant allele which can mask the small t allele. So small t allele is not expressed in this case. So those small t are called the recessive alleles. Recessive alleles are masked alleles by dominant allele. Masked allele is the small t. By dominant means that is a capital T. So example, small t allele is masked by capital T allele. So what happen in this? One is small t, one is capital T means this like this. That is in a heterozygous or hybrid condition. In this heterozygous or hybrid condition, what happen? The capital T and small t two alleles are present in that plant, but the plant is height by because the capital T is expressed, the small t is masked or not expressed. So that expressed character is called the dominance, which is not expressed or masked is called the recessive character. For example, small t. Dominance, for example, capital T. Recessive, for example, small t denoted. So another one is the segregation. What do you mean by segregation? Segregation is nothing but separation. Separation of alleles during gametes formation. Of course, the alleles are present on the two chromosomes at same locus and determine one character. But during the formation of gametes, these two alleles can enter into the separate gametes without influencing one another. That is called the segregation. So another one is the independent assortment. Okay, however, this segregation means the two alleles present on the two chromosomes. So two alleles present on the two different chromosomes at same locus and determine one character. But independent assortment is different. Means alleles of different traits. But in the segregation, alleles of same trait. But independent assortment, alleles of different traits located on same chromosome. Independently and separately enter into gametes. So here there is some difference in between the law of segregation and law of independent assortment. What are the differences? Means in law of segregation, the two alleles present on two chromosomes can enter into the gametes separately. That is called law of segregation means these two alleles which are present in that on that chromosomes can determine one character, but here in the independent assortment the two or more chromosomes which are present on the same chromosome side by side same chromosome and determine different traits also enter into the gametes separately and independently and enter into the gametes. This is called the independent assortment. We will discuss clearly. And then, so another terminal is the homozygous alleles. Homozygous alleles means those which are same alleles. Alleles of same type, for example, capital T, homozygous, and small t, homozygous. So these two are the homozygous alleles. For example, capital T is homozygous dominant alleles because they can express themselves. Small t are the homozygous recessive alleles. They do not during this means if two small t alleles are there, then it is expressed. Its character is expressed during the hybrid conditions or heterozygous conditions. The small t character is not expressed, that is masked by the capital T or dominant allele. So here we have to know the meaning of homozygous alleles means if the two alleles which can determine one character are same, those are called the homozygous alleles. If they are different, what happen? Yes, okay. Homozygous dominant in this. Homozygous dominant means that is a capital T tall or capital Y yellow in color. These are the dominant genotypes. These are in homozygous in condition same, and also those are the dominant characters. And heterozygous dominant means, for example, if your plant is capital T and small t, then what happens? Whether it is tall plant or it is dwarf or short plant means it is tall plant, because in a heterozygous conditions or hybrid conditions the dominant characters are expressed, the recessive characters are not expressed in this case. In this case also we can see capital Y and small y alleles are there. Capital Y can determine the yellow color, small y can determine the green color, but here we cannot see the green color in that seed because the dominant allele capital Y is there, who only the yellow color is expressed, the green color is masked or suppressed or not expressed in this case. So these small t and small y characters are recessive in this case. Capital T and capital Y alleles are dominant in them. So during the heterozygous condition, if the dominant allele is there, the dominant character is expressed in that organism. The recessive alleles are not expressed. So homozygous recessive means those alleles which can express themselves when they are in homozygous conditions. For example, small t, when do we get the dwarf plant? Means, for example, if the capital T is there, tall plant. If the capital T small t is there, in heterozygous condition also it is tall plant. But when do we get the dwarf plant or short plant? Means homozygous recessive alleles. If two recessive alleles are there, then we can get the short plant or dwarf plant in this case. If you want to get the green color seeds producing plant, then that plant should contain two small y alleles, then it can produce as green color seeds containing plants only. These are the homozygous alleles, same type of alleles. And homozygous dominant allele means those alleles which are expressed. Heterozygous dominant alleles means during the hybrid or heterozygous conditions also those dominant characters are expressed. Those are called the heterozygous dominants. Homozygous recessive characters are expressed when the recessive alleles, when the two recessive alleles are present. In that cases, these are the. And next, for one character or one trait, that Gregor Mendel conducted his experiment as the monohybrid hybridization for one character or one trait. So one character means in those 7 characters which he chose, from that 7 character he took only one character. For example, tall or dwarf plant, or purple color flowers or white color flowers, like that. In this case we will see flower color. For example, he chose the flower color. In this flower, that is purple color flower, uh, and another one is the purple color flower. Um, if two purple color flowers are made cross pollinated, then what happens? This is the F1 generation actually. We'll see first this one, or yes, this one we will take for example. Mendel took two plants here. One is tall plant, another one is the dwarf short plant. Pure tall plant and pure short plant. So the phenotypically it is tall, phenotypically it is short or dwarf plant. Genotypically it is capital T, pure lines. Genotypically it is small t, pure lines. So these two are homozygous alleles capital T, homozygous alleles small t. Then what happened? Uh, Mendel crossed these cross-pollinated these two plants, tall and short. The pollen grains which are produced in these tall plants are allowed to fall on the stigma of ovary in this plant. Then what happened? The pollen grain combined with the ovule and forms a zygote. From these zygotes, the next generation plants were developed. In these plants, this is called the parental generation, pure lines, uh, homozygous dominant, homozygous recessive, parental generation. Uh, then in next generation, this is the F1 generation. From in this F1 generation, he pollinated selfly. It means self-pollinated in these. Why? Because, for example, this is denoted in the checkerboard. Uh, this is tall plant. This is containing capital T and capital T. This is small t and small t. During the gametes formation, what happens? These alleles, these diploid alleles are separated and divided into haploid alleles. So what happened? The gametes are all the gametes produced from these plants are containing capital T only. All the gametes which are produced from this plant are containing small t only. So what happened? Capital T combined with small t and forms the capital T small t. Genotypes are all the genotypes produced in this F1 generation. F means filial, so means next generation. In this generation are all are tall plants. Why all are tall plants? There is no dwarf plant is produced in this F1 generation. Why? Because the plants produced in these are phenotypically all are tall, but genotypically these all are not same, because genotypically means these all are actually hybrid plants. All plants produced in this F1 generation are capital T and small t heterozygous condition only. Here we can say that already heterozygous dominant, one character we discussed here, heterozygous dominant. In this heterozygous dominant condition, the alleles are capital T and small t. So if the capital T and small t is there, capital T is the dominant allele, small t is the recessive allele. So what happen then? Capital T allele is expressed, small t is must not expressed. If the capital T is expressed, what happen? Automatically the plant will become tall. So in the F1 generation, he found that all the plants are tall. Then he questioned himself that, but where is the dwarf plant alleles? Um, he questioned himself, and self he pollinated themselves, self pollinated, because whether the recessive allele is present in this case, or what happened to that allele. So to test that allele, he self-pollinated these F1 hybrids. the gametes which are produced in this are from this plant capital T and small t gametes, from this also capital T and small t gametes. From this capital T and small t gametes, for example, yes, first plant you can see in the F2 generation, tall plant. So why? Because in this case capital T and capital T are produced. Next generation, sorry, in the next organism, next plant in the F2 generation, this is also tall plant, and third plant is also tall plant, but the fourth one is the short plant. He got three phenotypically three are tall plants, and phenotypically fourth one is the short plant. So here what happened, in the parental generation the dwarf character is present, but that dark character is hidden in the F1 generation, and it is again reappeared in the F2 generation. Means nothing happened to the dwarf allele in the F1 generation. That is just hidden by dominant allele or dominant genes, and later in the F2 generation automatically, without any disturbances, without any changes, that is freely expressed is its dwarf character. Means what I mean, this is called the law of segregation. Here we can find these two principles, law of dominance and law of segregation. So here, where is the law of dominance? Means here the law of dominance in during the heterozygous condition. These all are heterozygous hybrid plant. In this heterozygous hybrid plant, he chose only one character here, so it is called the hybrid. Monohybrid, mono one character, and it is hybrid, so it is called as the monohybrid hybridization. F1 generation plants are self-pollinated. From these self-pollinated, got two types of alleles, capital T alleles and small t alleles. Capital T are combined to form pure homozygous dominant capital T plant. One and two are, these two are heterozygous dominant capital T and small t. Heterozygous dominant, this is homozygous dominant capital T. These two are heterozygous dominants capital T and small t. Although they are tall plants, because dominant character is their capital T, but the fourth one is the small t, small t and small t homozygous recessive alleles. Two are the homozygous recessive alleles, since it is in dwarf case. So if you want to get the dwarf plant in the physum sativum, those alleles which are present on the chromosome should be recessive. Two should be recessive, then we can get the dwarf plant only. So the phenotypic ratio, if we in the F1 generation phenotypes, all phenotypes are tall in, okay, right. In the F2 generation phenotypic ratio, we can get 3:1. In the F2 generation phenotypic ratio is F 3:1, means 3 tall plants, 1 dwarf plant, 3:1. But the genotypic ratio is different, genetic makeup, because this is pure tall plant capital T. These two are not pure tall, because these are heterozygous dominant capital T small t. This is also capital T small t, heterozygous dominant two plants. One is homozygous dominant, 1:2. Two are heterozygous dominant, 2:2. One is pure dwarf plant, homozygous recessive plant, 1:1. So from this F2, from this monohybrid hybridization in the F2 generation, phenotypic ratio is 3:1, 3 tall plant and 1 dwarf plant, and the genotypic ratio is 1:2:1, means 1 homozygous dominant, 2 heterozygous dominant, 1 is the homozygous recessive plant. This is example. In case of seed shape, if you observe, capital R are the round color homozygous dominant characters. Small r are homozygous recessive characters. They produced gametes which are containing capital R alleles and the small r alleles. If they are denoted in the checkerboard, what happened here? Yes. no, not this one actually. This is homozygous dominant, this is heterozygous dominant. So this is F2 generation. This is actually monohybrid cross, but it is not started, it is middle. Okay, however this one we will see heterozygous condition. Okay, heterozygous, this is the F1 generation. Okay, capital Y and small y, capital Y, small y produce a capital Y are the dominant homozygous dominant, small y are the homozygous recessive alleles. These from these alleles the gametes are produced, the gametes are containing capital Y and some are small y. If the capital Y is combined with the small y, so that is cross pollination. You know what happen? Capital Y and small y all are heterozygous dominant seeds are produced which are in yellow color. So if the all are heterozygous dominant, likewise in this heterozygous tall plant, here heterozygous dominant is the yellow color. In between these heterozygous or hybrid plants, if they are self-pollinated, they produces capital Y and small y alleles. Those capital Y and small y alleles here, they are done. This is capital Y allele, this is capital Y allele, this is small y, this is small y. Then what happened? Capital Y. So homozygous dominant, yellow in color. This is capital Y and small y. Although it is yellow in color, but it is heterozygous dominant because one capital Y allele, one small y. Here small y allele which can determine the green color is not expressed, only the yellow color is expressed. So because it is the hybrid or heterozygous dominant, and this is containing one capital Y allele, one small y allele, same like that, heterozygous dominant. But the fourth one is different in this generation, F2 generation. This is small y and small y, so it is in green color, means just like our dwarf plant, fourth one. So the phenotypic ratio are 3 are yellow in color, 1 is green color, 3:1 ratio again we got. But genotypic ratio is different, 1:2:1, because only this color is homozygous dominant, capital T. These two are heterozygous dominant, capital Y and small y. This third one is the pure homozygous recessive allele, small y. So 1:2:1 genotypic ratio. From this is also okay, but here he started from F1 generation. In the F1 generation, all are hybrids because capital P and small p, capital P and small p. Parental generations are pure capital P, small p, small p. But in the F1 generation, as usual, hybrid plants are produced. Here, these two are hybrid plants actually, means heterozygous dominant. In the heterozygous dominant position, the purple color is the dominant, but white color is not appearing in this F1 generation. So white color is recessive character, purple color is the dominant character. So heterozygous condition, we can see capital P, small p, and this is also if these two plants are self-pollinated, alleles are produced. Those alleles, some are containing capital P, small p, capital P and small p. Okay, these from these in the F2 generation, these four plants are formed. In these four plants, he observed that three plants are phenotypically purple colored, purple colored flowers containing plant. Only one plant is there which was flowering with white color flowers, only one. This is the phenotypic ratio, 3 purple color, 3:1 white color, 3:1 ratio already we observed in the others. Phenotypic ratio is again 3:1, and the genotypic ratio, if we observe genotypic ratio, we can get the 1:2:1, because 1 is the pure capital P. Here you observe capital P means homozygous dominant purple colored flowers producing plant. These two are not homozygous dominant. They are heterozygous dominant because one is capital P dominant, one is the small p recessive. This is also capital P dominant, small p recessive. So these are the heterozygous dominant 2. 1 is to 2 is to 1 pure white color, small p, um, white color white color flowers producing plant is developed, produced. So phenotypic ratio in the F2 generation is 3 is to 1 again. Genotypic ratio is again 1 is to 2 is to 1. Okay, this is, and this is also same like that, but here the parental plant is crossed with the hybrid plant. This is we can get another ratio. So these three, from these three we observe that in the monohybrid hybridization, John Gregor Mendel chose one character in those any one in the seven, any one character in the seven traits. Uh, from that one character in the F1 generation, he got hybrid plants. These are the hybrid plants, heterozygous dominant hybrid plants. Those hybrid plants which are produced on in the F1 generation are self-pollinated. But why he pollinated? Uh, why he pollinated them in self-pollination means? But the question is, here in the parental generation, the dwarf allele is there, that is small t allele. Where is that small t allele? If these all plants produced in the F1 generation are tall, those tallness is determined by the capital T, or those yellow color is determined by the capital Y, or those purple color is determined by the capital P. But where is the small p in the F1 generation? Where is the small y? And where is the small t in the F1 generation in the hybrid condition or in the heterozygous condition? Means that is not expressed. Okay, that is not expressed. Whether it is there or not, it is escaped. So he self-pollinated the hybrid plants in the F1 generation. In the F2 generation, he got once again the dwarf plants. So he concluded that in this monohybrid hybridization, when he chose one character, the recessive character is hidden in the first generation, but nothing happens to that uh, recessive gene or allele that is expressed in the next generation or F2 generation without any influence or without any disturbance. Means in the F1 generation, capital T and small t alleles are located side by side, but they do not mix and they do not influence or they do not disturb. They are lying just side by side. The dominant character is expressed. The small character is just sit on the chromosomes in the next generation, that is freely expressed. So that is main thing. So here the two principles are law of dominance in the F1 generation hybrid condition. Here the law of dominance in the F2 generation. What happened? Those two alleles are segregating, are separating into separating and enter into the gametes without any disturbance. Those are called the law of segregation. So law of dominance is the expression of dominant character. Law of segregation is those two alleles present on the chromosomes are separated or segregated during the gametes formation. That is the that's in that manner the traits are transmitted to the next generation. That's why these small t character is again reappeared in the F2 generation. So in this manner, law of dominance and law of segregation, the parental characters are sometimes expressed, sometimes those are masked, and they can be reappeared in the next generation or in the next generation. Those are the inheritance. Those are called the Mendelian inheritance from the law of dominance and law of segregation. And the monohybrid hybridization, if you will see this, these are the pure lines in P generation, homozygous dominant and homozygous recessive, pure tall plant and pure dwarf plant. From this pure tall plant, um, during the gametes formation, the meiotic division take place. These diploid condition, 2n condition, the meiotic division produces haploid gametes. Capital all are containing the capital T because this is the pure tall. This plant can produce gametes. These are all the gametes are containing small t alleles because it is the pure torf. So, for example, if it is the male plant, if it is the female plant, the due to the cross pollination, the pollen grains are allowed to placed on the stigma of this ovary. Then what happen? Those male gametes are combined with the female gamete and produce the diploid again. Diploid um, plant. This diploid plant is different from this parental plants because this is capital T, this is small t, but this F1 generation plant is capital T and small t, two different alleles are there. These two, in these two, then what is the phenotype of this plant? Means this is tall plant, just like this, pure tall plant. If it is the tall, where is the small t character? Where is the small t factor? Where is the small t allele or gene? That is not expressed. Just that is there in this hybrid plant, but it is not expressed. It is not showing its character phenotypically or externally, but it is there in the F1 generation. This is the hybrid plant. This hybrid plant is produced by taking one character, so it is called the monohybrid. From the this is formed from the hybridization or pollination. Okay, however, to know the presence or absence of this multi allele, Mendel continues his experiment from this F1 generation to F2 generation. In the F1 generation, the gametes produced are some are containing capital T allele and some are containing small t alleles. Self-inbreeding or self-pollinated on this checkerboard. This is the male plant, for example, produces same type of alleles. Female plants also produces same type of alleles. From this, what happened? Capital T, that is pure tall plant, just like his grandparent. Capital T, small t, this is heterozygous dominant plant, like their parent plant, hybrid, heterozygous dominant. This is also parent, like parent, heterozygous dominant. These two are, and this is homozygous recessive plant, which is not visible in the F1 generation hybrid condition, but it is observed in its grandparents generation, small t, means this dwarf plant is short plant is produced when the two alleles are recessive only. That is the point here. The two alleles are in recessive condition only, this dwarf character or recessive character is expressed. Otherwise it is be just like the heterozygous. And if it is so, from these monohybrid hybridization, the phenotypic ratio is 3:1 because these three are dominant tall plants. This is short dwarf plant. So the phenotypic ratio means externally the three are tall plant, one is the dwarf plant. So 3:1 ratio, but genotypic ratio is one pure tall plant, capital T, 1 2 heterozygous dominant, 2 is to 1 homozygous recessive plant, 1. So in the monohybrid hybridization, this is the law. Why do the plants are tall in the hybrid condition? Means dominance of capital T. Here also dominance of capital T. If one capital T is there, that is automatically dominant character because it is the dominant allele. So 3:1 ratio. Genotypic ratio is genetical makeup is the 1:1 homozygous dominant, 2 heterozygous dominant, 1 is homozygous recessive. So during the gametes formation, capital T and small t alleles are segregated. Yes, during the gametes formation, these two alleles are segregating, are separating. That is the law of segregation, and enter into separate gametes. These two are separate gametes. This is separate gamete. This is called the law of segregation, law of dominance and law of segregation. From the monohybrid hybridization, Mendel proposed two principles. One is the law of dominance and another one is the law of segregation. Okay. Once again, if you will recall it, the law of dominance means in heterozygous condition, in F1 generation or in the F2 generation, means the we found heterozygous condition in F1 generation and F2 generation also. This is F1 generation, these all are heterozygous in condition. In the F2 generation, we found 2:1 ratio. These are the heterozygous dominant. In this heterozygous condition, that is expressing only dominant characters only. Okay, this in a heterozygous condition, in either it may be F1 generation or in the F2 generation, that is capital T and small t, or capital Y and small y. The capital T allele is expressed, so it is tall, but the small t allele is masked, it is not expressed. So that is the law of dominance. And another one is the law of segregation. In F1 generation, the masked character is small t, we know that, is again reappeared in F2 generation. Fourth one, you see this one, the masked character in the F1 generation is again reappeared in the F2 generation. So those are F2 generation without any disturbance. Okay, however, in the F1 generation, those are present. That is a dwarf character small t is present, but it is not expressing here. But however, in the next generation, F2 generation, that is reappearing, means okay, that small t or recessive allele is safe, but it is not expressing in the F1 generation. This allele is automatically expressed in the next generation. Why? Because means the alleles in the parents are hybrids, are segregated during gametes formation and enter into separate gametes without any influencing one another. If you will observe once, this is chromosome, this is another chromosome, this chromosome is containing capital A allele, this containing capital A allele. Okay, these are the just capital T and capital T. Imagine these two alleles are present in the two different chromosomes at same locus, which can determine one character. During the um, during the gametes formation, what happened? These two alleles present on these two chromosomes are separated and enter into separate gametes. That is called the law of segregation. For example, in this condition, we'll see. For example, capital A. Imagine capital T. This is small t. This plant is tall plant because this capital T allele is there. So this is called the law of dominance. This is the separation of these alleles during the gametes formation is called the law of segregation. So that is monohybrid hybridization, and another one is the dihybrid hybridization. In this, okay. However, we will discuss about the dihybrid hybridization in the next video classes. Thank you.